All right, well, welcome back, everybody. Shh, please. Today's session 9, Pathways. We're kind of building up in levels of organization. We started with atoms, then molecules, which are made of atoms, proteins, which are made of amino acids, proteins are enzymes, and now enzymes get organized into pathways, which we'll talk about today. There's a couple fun examples I want to do. There is, as usual, a pre-lab due next week. Your take-home exam is due tonight, 11:59. Before we go further, questions about exam number 2? If people are having trouble, yes. Oh, that's okay. If people are having trouble with the screenshot part of exam 2, where you got to get your little name taggy thing from Blackboard to line up, the secret to doing that is opening Blackboard in a separate browser window, not a tab, but actually ask, for a new window, because then the window you can slide around any way you like, and you can set it up in the background, and that'll be super spiffy. So yeah, make sure you use a different browser window, and you should be all set. Set up for exam 3, which is apropos of what you had in lab this week. So there are 2 parts of that. I want to talk about the cone of silence briefly. Oh, yes, the cone of silence. When in the cone of silence, you may speak with Brian and your TAs. Awesome. All right. So there's 2 parts. You're finding interactions between amino acids, side chains. You're doing some amylase, which is what you did in lab. Finding the interaction, taking screenshots, totally fine to help with you, work with your buddies on that. You did that in lab. Drawing your own structures, writing up your answers. That's what I want to see you show what you can do. And then there's interactions with another protein, subtle lysine. And that one, that's all you. Right? It's a very much exactly what you did in lab, except with a new protein, your own a bunch of amino acids. I want you to show me what you can do on that one. So that whole part is in the cone of silence. Are there any questions about that? And that exam will be due next Friday. Other questions of a logistical or administrative nature before we get down to business? Very well. Hearing none, I will proceed. So this is a little bit of a reiteration, but I just want to make sure we're absolutely on the same page, because we're going to be doing a lot of things like this. So we're going to see a lot of diagrams like that, so I want to make sure everybody's on the same page what they mean. So take a second, look at that. What is XACE? So I'm going to talk about it. So we'll come around. Let's do one more minute on this one. All right, 10, 9, 8, 7, 6, 5, 4, 3, 2, 1, 0. All right. So a BC split. It is, yes, an enzyme, but there's actually a better answer, which is the enzyme that catalyzes that particular reaction. Right? So yeah, C is technically correct, but this is the enzyme It speeds this reaction. All right? What do we call A and C? Pardon? They're not necessarily amino acids. Yes, somebody said substrate. Yes, that's right. There's a bunch of different things you can call them. They're small molecules, but these guys are substrates. And you must have both or no reaction. All right? That's an important thing. That is, enzymes are real fussy. They only do one reaction, typically, and they require exactly the right conditions. So if you had just A in the enzyme, nothing happens. Just C in the enzyme, nothing happens. You've got to have A and C and the enzyme for the reaction to happen. Okay? That's important when we think about what happens, because there's going to be a lot of things we're going to talk about when certain molecules are missing, what are the consequences of messing this up. So understanding that's important. And the other thing about these guys, B and D, we call those products, yes. And you always get both, or neither, if the reaction doesn't happen. So you always get B and D, right? Questions about that, because we're going to use these a lot today and then going forward. All right. And the X-ACE, typically, but of course not always, because science is kind of weird that way, enzymes' names end in ACE. All right? Phenylalanine hydroxylase, RNA polymerase, that sort of thing. And we don't draw their structures typically because they're really, really big and complicated. They're this big blob that does the reaction. But we will typically draw the structures of the substrates, at least some of them, and the products, because those are sort of relevant to the story. All right. If we were going to translate this into English, we would say that X-ACE binds A and C at the active site, converts them to B and D, and releases them. That would be the English translation of that diagram there. All right. So what I want to talk about for the rest of the class is two for instances. The first one is about vitamin C. Because there's an interesting enzyme story behind that one. So, here, you tell me, why do we call vitamin C a vitamin? What makes a thing a vitamin? Let's do a minute more. Let's do 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. All right, where are we at here? All right, so ABC, strong for B. Let's take a look. It is necessary for a healthy life, but it also turns out we can't make it ourselves. That's the B is the most right answer, right? Because there are some things that are necessary for a healthy life, but that we make, like blood, for example, which we make ourselves. All right. Again, this is why I hate multiple choice questions. It's mostly to get people thinking about these things. Necessary for all living things. Does anybody know what other animals, do all other animals require vitamin C, or only some? Does anybody know that? Pardon? It turns out no. So it turns out, like those adorable cats that everybody squeed over on Wednesday, there's no vitamin C for cats. They make it themselves. I mean, they need ascorbic acid, but they can make it. Dogs, cats, most other animals. Humans, other primates, these sort of monkeys, apes, gorillas, that kind of thing, they need vitamin C in their diet. They need it, and they can't make it. And also, this really obscure guinea pigs, apparently, need vitamin C and can't make it. But all other animals, there's no such thing as vitamin C for cats because they make their own. So it's not a thing you have to give them. Right? So it's kind of weird why some animals can and can't. We'll talk about why in a minute. But with me on this about what it means to be a vitamin. Cool. All right. So what I want to do now is address your attention to the part of today's information packed handout. So ascorbic acid is also known as vitamin C. All right. So this is how vitamin C gets made in some living things. Almost all pathways start with glucose. That's where the carbon atoms come in. There's 4 enzymes, 5 steps in this pathway. There's an enzyme, glucose dehydrogenase. Glucose is its substrate. Glucuronic acid is its product. Glucuronate reductase takes glucuronic acid as a substrate, makes gulonic acid as a product, et cetera, et cetera. This last step is kind of weird. Turns out 2-keto blah, 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 on its own, if you just put it in water, doesn't need an enzyme. It will turn into ascorbic acid, otherwise known as vitamin C. Right? The other ones, so that's the point about enzyme and all these chemical reactions. They're all possible. All of these happen, but they happen so slowly we would never get enough vitamin C to live. So we need enzymes to speed them up. Turns out this one reaction is fast enough on its own that it doesn't actually need an enzyme. That's pretty unusual in biology, but it turns out to be an interesting thing here. Right? Every one of these other reactions, like I said, is possible, just happens way too slowly to give us the vitamin C we need. Questions about interpreting that, the difference between like gulonolactone oxidase and gulonolactone, which is its substrate. I'm going to share this with me because we're going to be thinking about this a bunch today. Okay. All right. So I want to ask some questions about this. So that pathway of synthesis of vitamin C, what do you think, based on what we just said a few minutes ago? You tell me. What's the deal on this? All right, let's go another minute on this one to 2:30. All right, 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. All right. Yes, because citrus fruits are noted for a high vitamin C content, they have all the 4 enzymes, and humans lack 1 or more. That's right, because we can't make it, they can. Likewise, what that also means is that my cats have all 4 enzymes, dogs have all 4 enzymes, but things like monkeys and gorillas don't. Right? Cool. So what enzyme or enzymes are we missing? It turns out that humans are unable to make gulonolactone oxidase. The gene is heavily mutated. All right. So therefore, we must, that's where it changed color on me. We must get vitamin C made by something else. Sometimes another organism, or it might be made chemically. And we eat vitamin C, not the enzyme. Because the enzyme would be digested in your stomach. So taking pills of gulonolactone oxidase wouldn't do any good because it would go in your stomach and get digested, so it wouldn't help you at all. All right? So questions about that one? Yeah. No, it's not that humans don't digest vitamin C. Humans don't produce vitamin C. Or, let me say it carefully. Humans don't produce ascorbic acid. That's why we have to eat it. So it's not. So let me show you on the pathway what we mean. So if you go back to the figure on today's information packed handout, this enzyme is missing in humans. I'm not sure what cells in the body do this, but somewhere in your body right now, there's a cell turning glucose into glucuronic acid, and that into gulonic acid, and making gulonolactone. And then it stops right there. So some gulonolactone is piling up in cells somewhere completely uselessly. And it's kind of weird. Why bother going through all those first 3 steps if you're not going to do the 4th? That's one of those weird things about life. So we cannot make ascorbic acid, which means we must eat it, which is what makes it a vitamin. In my cats, they have all of these enzymes. They make ascorbic acid, so they're all set. It's not a vitamin for them. It's a thing they need, but they make all that they need. Questions about that before we talk about some more practical applications? Yeah. Pardon me? Why isn't vitamin C broken down in the digestive system? That's an interesting question. Presumably we lack specific enzymes that would do it. And the other thing is, for what it's worth, vitamin C is not sensitive to acid. It is an acid itself, and so it's stable in stomach acid conditions. Which is also fortunate, I suppose, now that you mention it, because if vitamin C was destroyed in your stomach, then you'd really be in trouble because you couldn't make it and you couldn't eat it. Right. That's interesting. All right, other questions? All right, so let's take this a little further. All right, so that's kind of a bummer. Are there any other things on this list that we could eat instead of vitamin C? Suppose you wanted to go into the supplement business and you didn't want to, the market's been cornered on ascorbic acid. Could you sell people pills of any of those things that would do the same job, would make the ascorbic acid that we need? So think about that one, talk to your neighbor, et cetera, et cetera. Let's do 30 seconds on this one. All right, 10, 9, 8, 7, 6, 5, 4, 3, 2, 1, 0. All right, so people say gulonolactone should work. So let's take a look and think about that one. So remember the goal. We got to get to ascorbic acid, vitamin C. If I took a gulonolactone pill and I don't have that enzyme, what's going to happen to that gulonolactone? Nothing. Absolutely nothing. Because without this enzyme, you can't skip steps. Without this enzyme, that gulonolactone gets turned into nothing else. It just sits there. So what would happen is you'd pass it out in urine eventually, right? It wouldn't do you any good. That any of those four molecules, they're before where the blockage is. So it's kind of like, that analogy is popping in my head. It's not going to work. But in any case, it's once you can't get past this step. So this one, even though it's right before it, it's so close. It doesn't really matter because without that enzyme, there's nothing to take that gulonolactone on to the next step. Right? So the majority answer was wrong. I want to be clear. The right answer is none of the above. None of those ones can do it. But there was significant dissent. So let's make sure that we clear that up. Talk to me. Are we cool with that answer? All right. So basically the answer is this. None of the above without All right, so let me ask you a question then. Is there any molecule in that pathway that you could package up into a pill that would work approximately as well as vitamin C? the 2-keto-gluconolactone. If you could, so for example, if you could package up this stuff into a pill, the 2-keto-gluconolactone, it will turn on its own into vitamin C. Now, practically speaking, it's probably really hard to make this stuff, but probably as soon as you make it, it turns into vitamin C. So you might actually really be able to make a pill of that stuff. But if you could, that would be fine, right? Because it's after the block. Right? Everybody with me on that one? It's important to get those pictures out. Yeah? So what is it that turns this into vitamin C? Looking at it, it probably reacts with water in a catalytic way. When you guys, if you go on and take organic chemistry, you'll probably look at that and go, oh, that's an enol reaction. It's a kind of thing that it's a common organic chemical reaction. It just needs water to do its thing. Other questions? All right. So I'm going to skip the next question because it's actually not that interesting and it ends up being sort of more confusing than strictly necessary. So we'll just nix that guy. All right. So I want to now switch gears. So we're done talking about vitamin C. I want to talk about a different pathway that works in a different way. So I need you to address your attention to the other figure on today's information-packed handout. That one. So this one, all right. Shh, I need this intro, please. The goal of this pathway we just finished about was to make vitamin C. You don't got it, you want to make it. The goal of this pathway is the reverse. You got something you want to get rid of. Too much of something, how do we get rid of it? Right? So they're similar, they're pathways, but they have a different goal. Right? So here's the deal. Phenylalanine, that's my favorite go-to hydrophobic amino acid. That's its structure. It's got its hydrophobic side chain. A sort of normal human adult diet, you eat about 150 grams of protein a day, roughly speaking. You digest that into amino acids. About 2 grams out of those 150 are phenylalanine. So it's actually not very common in proteins, but it's there. So you eat about 2 grams of phenylalanine a day. That's its structure. It turns out as an adult who's not growing or bulking up or anything, you need about 0.5 gram of phenylalanine every day just to make new proteins, replace things that got worn out, whatever, just maintenance stuff. What that means is if you eat 2 and you only need 0.5, you got 1.5 grams of phenylalanine you got to get rid of. And interestingly, these are not like calories. Like if I eat more calories, more sugar or fat than I can use, I store it as starch or fat in my body. But animals, in general, cannot save amino acids. I'm not really sure why that is. It seems that's actually sort of a pain that we can't. In any case, we can't. So that extra 1.5 grams of phenylalanine has got to go somewhere every day. And in the end, it goes out in urine. That's the most common place we get rid of stuff. Urine is mostly water. Phenylalanine is not the most hydrophilic molecule I've ever seen. It's not going to dissolve very well in your urine. So what your liver does is it breaks it down into molecules that are much more hydrophilic, so they can be passed in urine. And the first step is it turns phenylalanine into tyrosine. And you can see it's an OH added on, so it's a little more philic. This is an enzyme called phenylalanine hydroxylase. This one we're going to spend a lot of time talking about, PAH. Then there are other enzymes that further transform it and make it more and more philic and bust it up into little pieces, and it goes out in urine. So the idea is, I've got this extra phenylalanine I don't need, and we'll see later on it's bad news if it sticks around. So this is a pathway for getting rid of something you don't want. It's kind of the opposite of up here, which is making something that you need. Right? So everybody with me sort of on the details of your phenylalanine budget, what this is trying to accomplish, and how that's different from that before we dive in. All right, so let us address ourselves to a question. And I apologize for the alphabet soup today, but the three-letter abbreviations are sort of useful. There are humans who are born without the ability to make that enzyme, PAH, that first enzyme in the pathway that causes a disease called phenylketonuria. It's a genetic disease. We're going to talk about lots of genetic diseases and how they work later. This is the first example. So people who can't make PAH, can't make the enzyme, have a disease called phenylketonuria, which we will call PKU. So first step is, if you are a person who cannot make that enzyme, what would you expect to build up in people who eat a regular diet and don't have that enzyme? So think, this isn't about what's going to get made, it's about what's going to build up because of a block. So think about that one, talk to your neighbor, et cetera, et cetera. Let's do one more minute on this one. Let's do 10, 9, 8, 7, 6, 5, 4, 3, 2, 1, 0. All right. Yes. It is the molecule So the idea is the following. If you block this guy, anytime you block a place in a pathway, the things after it don't happen. And the thing right before it piles up. So in us, there are piles of gulonolactone waiting for an enzyme, waiting for their prince who's never going to come. Sadly, they're not reacting. Likewise, in phenylketonurics, phenylalanine piles up because there's no enzyme to take it that next step and break it down. That was a strong consensus. I want to make sure everybody's with me on that before we go further. All right. So let me write up what the consequences of that are, because that leads to disease, it turns out. So it turns out here that a PKU eating a normal diet leads to very high phenylalanine in your blood, which leads to brain damage. All right? And so it turns out Now I think they use different tests, but there was, this is a relatively rare disease, but it's a rare disease, but it's very consequential. If you're an infant and you're a phenylketonuria, you would typically get a normal diet. Your parents wouldn't notice anything was wrong, except there's accumulating brain damage, and then that, especially to a developing brain, is particularly bad. So there are some neonatal tests that are done sort of basically right away when a little kid's born. Typically, they used to take a drop of blood from their foot or their little toe and test it to see whether there are high levels of any of these things so that you catch it early. And we'll talk about treatment in a second. But it has a very serious consequence because of the excess of that amino acid in your blood. Right? And so phenylalanine builds up because that's the one right step beforehand, and high levels of that can be particularly bad for you. Questions about that before we talk about treatments and things like that? Cool. All right. So my next question, what could you eat? What, if anything, could you eat to make it better? Let's do another minute, everybody. All right. Let's do 10, 9, 8, 7, 6, 5, 4, 3, 2, 1, 0. All right, we've got a BE split. So it's either none or tyrosine. All right? So, is the problem here that we're not making urine or that phenylalanine is piling up? Phenylalanine is piling up. So if you eat lots of tyrosine, is that going to help with the phenylalanine piling up? No. It won't do you any good. I mean, it won't necessarily be bad. Your urine will be a little more rich, but it won't do anything about the phenylalanine, all right? Because you can't sort of pull on a pathway, right? If you don't have that step, it's not going to work. There was significant descent. I was right up. So the answer is actually none of the above, right? Because but as I saw, there was significant descent. I want to make sure we're cool on this because it's weird. It's sort of ish backwards from the vitamin C thing, right? But that's because the goals are different. In vitamin C, you're trying to make vitamin C. And in this, you're trying to make the phenylalanine go away. Right? And so they've reversed that way. So when you're thinking about how pathways work, you want to think about sort of what the goal of the pathway is. Questions about that one? Because there's two more I want to do carefully. Okay. All right, so, well, and also, so based on all of that, suppose you were a phenylketonuria, someone missing that enzyme. How much phenylalanine could you eat each day and still live and not get brain damage? So think about that one. Talk to your neighbor. All right, let's do another minute. All right, 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Here we go. So an AC split. All right. A, 0. Could there be a problem with that? Why would there be a problem with A, with 0? You still need phenylalanine. So yes, it's bad news in large amounts, but you also need some. These are definitely not good because you need some 0.5 grams per day to live. So C is the right answer, right? And so the thing is, so there's a couple things about that. So if you are a phenylketonuria, you can live a normal life if you eat a very carefully controlled diet. Basically, they make, this is when, I don't know if you remember was it the summer and the spring, there was a trouble with infant formula. One of the big problems with infant formula is the kids that need special infant formula was almost impossible to get. So basically, if you're a phenylketonuria, you get regular, so it's like a protein shake, but with just enough phenylalanine to get by, because any extra is going to cause you problems. And so that you don't get to eat a delicious diet for the rest of your life, but you can live if you watch your phenylalanine intake very, very carefully. And the reason, if you get a diet soda with aspartame, this basically one can is about 0.09 grams of phenylalanine. So the aspartame contains phenylalanine. And one can of diet soda is about 1/5 of your daily phenylalanine budget. So if you didn't know that, you could get in serious phenylalanine trouble if you were a phenylketonuria. So that's why there's that warning on that thing. Even though people are relatively rare, it could be quite severe. So, or you'd have to plan sort of all day what you eat so you can have your can of diet soda. Yeah. Can you not? Right. Wouldn't it be nice if you could give them that enzyme? The problem is if you eat the enzyme, it's a protein. It's going to get digested in your stomach. If you inject it, it would only last a certain amount of time, and it has to be in the right place. So we'll talk more, but what you'd have to do is mess with people's genes and fix the broken gene is basically the only way to do that. Questions about that one? All right, last question I want to ask, and I want to say a little bit about it at the end, so don't be rushing off yet. Make sure you get your money's worth. What about the rest of us? Anybody in here with the phenylketonuria uric would know. So, and most likely nobody in the room is. They're really quite rare. So what about the rest of us that don't have phenylketonuria? What do we know about how much phenylalanine you can consume in a day and be okay? All right, let's do 30 more seconds on this one. Hang on, we're not quitting time yet. All right, 10, 9, 8, 7, 6, 5, 4, 3, 2, 1, 0. All right. Ooh, it's a DE split. All right. So, the argument for 1.5 grams makes sense. We know that that's going to work. Somebody give me an argument for E. Why would you say E? Somebody, why would you say E? Yeah. Oh my goodness, you're right. I did the math wrong. All right, so what I meant is excess. You're right, of course, because you can eat 2 grams because 1/2 gram you need and 1.5 you can throw away. Oh, I actually meant E as literally worded. I never thought, thank you, I never thought, I goofed up the question. All right, so yes. That's right. Just because you can deal with 2 doesn't mean you cannot deal with more. We don't know what the max is. That's the point I was trying to make. So we probably don't have to worry. I mean, if you got like a huge bowl of phenylalanine, it would probably taste nasty, and you might push the limit of things. But the main thing is we don't actually know. Thank you for pointing out about the math. I got to fix that for next year. All right, everybody, good job. Have a good weekend, and I'll see you on Monday.